Good morning students. In this video we will discuss a very small but very important topic of genetics, that is incomplete dominance. So what is incomplete dominance? So before discussing this incomplete dominance, try to recall the monohybrid cross which was performed by George Mendel. So in the monohybrid cross, so what Mendel did? So he crossed pure tall plant with pure dwarf plant. So what is the meaning of pure tall and pure dwarf plant? So pure tall plant means having the genotype capital T, and pure dwarf plant means having the genotype small t. So simply Mendel he crossed pure tall plant with pure dwarf plant, and what result he obtained in F1 generation? So in F1 generation he obtained that 4 progeny. So out of these 4 progeny, so all the 4 progeny which was produced during the F1 generation, so they resemble either of the one parent. So all the 4 progeny, so they resemble with the tall parent. So none of the 4 progeny they resemble with the dwarf parent. So it is very important point. So all the 4 progeny they resemble with either of the one parent, and which one that parent? So the answer is the tall parent. But in case of incomplete dominance, so when experiments on peas were repeated using other traits in other plants, it was found that sometimes the F1 had a phenotype that did not resemble either of the two parents and was in between the two, and this is actually the incomplete dominance. So in case of incomplete dominance, what will happen? Normally what happens that the F1 progeny which will be obtained, so it will resemble either of the two parents. But in case of incomplete dominance, the phenotype did not resemble either of the two parents and was in between of the two. This is the incomplete dominance. The inheritance of flower color in the dog flower, which is also known as snapdragon flower or antirrhinum, is a good example to understand incomplete dominance. So this phenomenon of incomplete dominance we can see in snapdragon flower. What is the another name of this snapdragon flower? So answer is antirrhinum. So this is a typical snapdragon flower which will show incomplete dominance. It's not like that all the plants they will show incomplete dominance. There are selected plants which will show incomplete dominance, and this snapdragon flower is one of them. So mainly in case of incomplete dominance, what happened? So in a cross between true breeding red flowered snapdragon flower and true breeding white flowered snapdragon flower, so a cross was made and the F1 progeny was pink. So so actually what happened in case of incomplete dominance in snapdragon flower? So there are different varieties of snapdragon flower. So that red flower, that snapdragon flower with red flower. Second one is a snapdragon plant with white flower. So the flower color in snapdragon, it is denoted by a gene, and this gene is commonly denoted by R. So what is the genotype of this red flower? Capital R, capital R. What is the genotype of this white flower? Small r, small r. So when a pure red flower possessing snapdragon plant was crossed with pure white flower bearing snapdragon flower, then in F1 progeny, the plant, that snapdragon plant which was produced, so they possess pink flower. So in Mendelian monohybrid cross, the F1 progeny, they they show resemblance with the tall plant, or they show the trait of the dominant parent. But in case of incomplete dominance, what we are seeing that the F1 progeny which was produced, so they neither show the red flower nor they show the white flower. So the F1 progeny they show a intermediate phenotype, and in the intermediate phenotype is pink color. So this is the incomplete dominance. So when the F1 was self-pollinated, when the F1 plant, whatever plant which was produced during the F1 generation, so when these F1 generation plant, so they were self-pollinated. So the resulted in the following ratio: 1 is to 2 is to 1. So the genotypic ratio as well as the phenotypic ratio is 1 is to 2 is to 1. So here, so when the selfing will be done, in order to get the plant of F2 generation, what are the different plants which will be produced in F2 generation? So what we do, so we mainly do self pollination of F1 progeny. So when selfing was done, then the result was, the phenotypic ratio was 1 is to 2 is to 1, as well as the genotypic ratio is 1 is to 2 is to 1. So simultaneously we will compare with the Mendelian monohybrid cross, what phenotypic ratio and genotypic ratio Mendel got in the F2 generation of monohybrid cross. So the genotypic ratio there was 1 is to 2 is to 1. So here also, in case of incomplete dominance, the genotypic ratio was 1 is to 2 is to 1. So only difference is regarding the phenotypic ratio. So try to remember the phenotypic ratio in F2 generation of monohybrid cross performed by Mendel. So the phenotypic ratio there was 3 is to 1. But in case of incomplete dominance, the phenotypic ratio is 1 is to 2 is to 1. So regarding phenotypic ratio, there is difference. So here the phenotypic ratio is one plant having the phenotype of red flower, two plants having the phenotype of pink flower, and one plant will possess the white flower. So if the allele combination will be capital R, then it will produce red flower. If the allele combination will be smaller, then it will produce white flower. But if the allele combination will be capital R, small r, so in that condition it will produce neither red nor white. So it will produce intermediate phenotype, that is the pink flower. So this is known as incomplete dominance. So in case of incomplete dominance, neither of the two, neither this capital R is completely dominant over small r, nor this small r it is completely recessive over the capital R. So that's why in F1 progeny, the genotype capital R, small r, it will produce a pink flower. So here the genotype ratio were exactly as we would expect in any Mendelian monohybrid cross. In case of incomplete dominance, so the genotypic ratio was similar to the Mendelian monohybrid cross ratio, but the phenotypic ratio had changed from the 3 is to 1. The 3, in case of monohybrid cross performed by Mendel, the phenotypic ratio was 3 is to 1, but in case of incomplete dominance, the phenotypic ratio is 1 is to 2 is to 1. So what happened was that capital R was not completely dominant over small r, and this made it possible to distinguish capital R, small r as pink from capital R red, and small r white. So this is all about the incomplete dominance. So what are the different questions can come from incomplete dominance? So first question will be, in which plant we can see the phenomenon of incomplete dominance? So answer should be snapdragon flower. What is the another name of this snapdragon flower? So it is also known as antirrhinum. What is the genotypic ratio in F2 generation of incomplete dominance? So answer will be 1 is to 2 is to 1. What is the phenotypic ratio in F2 generation of incomplete dominance? So here also the answer will be 1 is to 2 is to 1. What is the phenotype of flower color in F1 generation when pure red flower will be crossed by pure white flower? Then answer will be, all the progeny of F1 generation, they will produce pink flower. Define incomplete dominance. What is incomplete dominance? So incomplete dominance is a phenomenon in which neither of the two genes will be completely dominant, and the F1 progeny, it will show intermediate phenotype between the dominant and recessive trait. So this is known as incomplete dominance. So this much only in this video. In next video we will discuss about the another important topic of genetics, that is the codominance. So thank you, have a nice day.